Hey guys, before we get into the nitty gritty of microbes and what we know today about bacteria and viruses and so on, I'd like to lay a bit of a foundation and a historical perspective, starting with early notions of disease. In the Bible, there is mention of quarantining those suffering from leprosy. Now, this is a bit ironic, especially considering coronavirus today. Leprosy is a slow progressing disease, and the chances are that the signs and symptoms of leprosy were not apparent. So probably leprosy was passed to several people before they were ever quarantined. But there is this notion that there was a transmissibility, that you could transmit this disease from one person to another. In around 500 BC, ancient civilizations used to think there was a supernatural cause for disease, but that it was carried in the air somehow. So the notion that there's something in the air, that was there, but they thought it was a supernatural cause. There was bad air. Ancient civilizations, including Greek and Roman civilizations, in Greek it was mal aria, so bad air, which of course is where we get the name for malaria, right? So another term they would use for this was miasmatic odors. Miasmatic odors that might be basically foul odors that might be found from swamps, and they thought this was an evil spirit, again, supernatural. Okay? Now, it's not that they were not scientific about this, because a Greek physician, Galen, would actually record and report the imbalance of fluids as a result of disease. These fluids were blood, phlegm, black bile, and yellow bile. So there was somewhat of a scientific approach and a recording of how disease affected people, but the cause was still a supernatural cause. Now, slowly, as time progressed, philosophers, including Hippocrates, Thucydides, and Varro, started to come up with new notions, new ideas about what caused disease and how disease was transmitted. Hippocrates was the father of Western medicine, and he believed that inhaling bad air could cause disease. Now, Thucydides, in the second image here, Thucydides was a father of scientific history. So he took this a bit further. The idea of evidence-based analysis, having evidence for what causes something. A cause and effect reasoning is something that Thucydides wanted to apply to the notions of disease. He also recognized that survivors of disease would not become reinfected when caring for those that were diseased. So this idea that there was some kind of an immunity gained by individuals that beat an epidemic was also there, even though here again we're talking as early as 500 BC to I think it was in the single-digit BCs, okay, that these individuals were alive. Now, let's see here. When we look at what some of the ancient civilizations actually did to combat disease, is recognizing that there's something in this foul air. The Romans actually had this elaborate system of aqueducts, right? And these aqueducts would not only bring in clean water, but there was this enormous sewage system that they set up called Cloaca Maxima, so just a big sewage system. And this would actually flow into the Tiber River. It would remove the foul air, something that might cause disease. They recognized there was something there going on. All right, and we have an image here actually of part of their sewage system. It's not very private, is it? We have no stalls here, right, for people to deposit their waste. Okay. Now, let's zoom forward a little bit here to a point where we could actually see something with a very rudimentary microscope. This microscope was designed by Anton van Leeuwenhoek, okay? And van Leeuwenhoek actually was a custodian, and in his free time, he was a scientist. And he was quite an excellent scientist in one major respect. He was very good at recording everything he saw, drawing everything he saw with this rudimentary microscope. This actually is the lens here. And then the specimen would be placed here on the end of this, the tip of this needle here, okay? And now Leeuwenhoek, he examined everything he get his hands on. He examined scrapings from his own teeth, rainwater. He even examined his own feces. And he would find these little tiny, what he thought were little tiny animals. And he actually coined the term animalcules, right, for these tiny little critters. So finally we were given the opportunity to see some of these things in the mid to late 1600s. Now remember again that the prevailing thought was that there are supernatural forces, this bad air that could cause disease, and people weren't completely clear on how it is that life would arise either, okay? So the notion that organisms would appear, even when take ourselves away from the idea of microbes, but just think about frogs and fish and mice, there's this prevailing idea of spontaneous generation, this idea that life can or could arise from non-living matter. So people throughout history would say, well, they found frogs or fish in pools of water that were separate from the river. And because they found frogs or fish in these pools of water, that they could arise from non-living matter. They just needed some kind of life force. People would talk about a vital heat. Aristotle talked about a vital heat that needed to be present for life to arise, or life force, okay? People would find mice suddenly where grain is stored. Now, remember, if you don't have the technology to record the fact that these frogs or fish were initially laid as eggs in these pools of water, or that these mice actually found their way to the sustenance of grain where the grain was stored, then nobody knew, right? Another idea that people looked at was the idea that something would grow on food. So to find maggots or flies on meat was a mystery to people. That somehow maggots would suddenly appear, but you can't see the eggs, right? So it may not be completely clear. So not only was there this idea that there was a supernatural force causing disease, but people also didn't understand exactly how life would arise with some of these kinds of things. Of course, today we know that life arises from other living things. Sperm and egg come together to make a new human, right? That eggs can be laid in the ocean, and from that you might have fish arise, right? Because then another fish would come along and fertilize those eggs. So we have Going into the late 1600s, we have van Leeuwenhoek identifying small animal-like critters, and the question is being asked, where do they come from? Okay? Remembering that before this, everyone thought that it is possible for life to arise from non-living matter. Okay? So this led to a bunch of experiments, all right? Now this particular experiment was done by Francisco Redi in the late 1600s. This was after some of van Leeuwenhoek's experiments, and he was able to show that with an open container with some kind of sustenance inside, like a piece of meat, that you can have the formation of maggots and flies within that jar, okay? He also showed that if you sealed the jar and allowed time to pass, that there'd be no maggots or flies on that meat. So this kind of agreed with this idea of bad air, right? There's something in the air giving rise to not only disease, but here now we see life, okay? So you see they're starting to develop a connection between where this life is coming from and disease, okay? It wasn't thought to be a living thing that caused disease. It was some supernatural force before. But now we see that there's something in the air that gives rise to life on this piece of meat, right? So there was a whole host of spontaneous generation versus biogenesis ideas. This idea was experimented with by these three individuals, Francisco Redi, Needham, and Spallanzani. Okay? Francisco Redi would find that there would be no maggots on meat when it's sealed or covered with a fine net or gauze. Okay? So no maggots, no life. Okay? If there's no net and it's left open to the air, there would be life ultimately given time. Now, Needham came behind Redi in the early 1700s, and he would boil a nutrient broth, so it would have animal and plant substances in the water. Okay, so think of it like chicken soup, maybe, right? He would boil this, and then he would allow it to sit and seal that flask, that container, to see if there would be growth. Okay, so he found that when he boiled this broth and placed it in a flask, he would eventually have microbial growth, even when sealed. Now Spallanzani came behind Needham and said, well, I think that Needham had some poor technique in that he didn't boil the broth long enough. So he came behind him and did some of the same experiments looking at boiled and sealed containers, and found that there was in fact no microbial growth. So this is kind of sealing the deal that there's something in the air that's giving rise to this life. Okay? Now, if we zoom forward to some of the work by someone you all have heard of, Pasteur, and you have heard of maybe pasteurization. Well, he came up with a really fancy kind of container, okay, where you could actually boil a nutrient broth, something that would give sustenance to living things, and then he would melt and mold the neck of that flask so that through gravity, whatever is in the air that might cause growth or cause life to appear would not be able to fall into the actual flask, right? So you could have some living things here. You could also have air, though. Let's see here. You could also have air pass through, because living things need air, right? Need oxygen, okay? But anything that happened to be living that was in the air would get caught in this little loop of the neck of Pasteur's flask, okay? Now, if he were to break the flask, like you see here, ultimately he would find that there would be growth. Okay. They also did some experiments after this where they actually took some of the contaminated fluid in the loop of the neck, added that to the broth, and it would also cause growth. So this idea that something fell into the flask when broken this was kind of an eloquent experiment done by Pasteur to show this. Now this is zooming forward into the 1800s, by the way. Okay, so this idea that disease was caused by a living thing, a living microorganism, still people were unsure of that going into the 1800s. Okay, but we're kind of developing more evidence to show that there might be something there in the air. Now, before we leave this idea of the Pasteur's flask here, I want to just kind of talk a little bit about the gravity of all of the work that Pasteur did. Pasteur had a major impact on microbiology. He believed in the application of science to help the people. He's a fascinating man. I read a book on the history of microbiology, and they talk about Pasteur as being a rather pompous individual, but he certainly did a lot for the field. Along with this idea that he believes science should be applied to help society, he helped brewers in Germany in his lifetime. Brewers in Germany were brewing beer and other alcohol, and they were finding that some of their vats, some of their large vessels of beer would go bad. And Pasteur thought that and identified the fact that some microbes might be growing within certain of those vessels of beer, microbes that you don't want to have in those vessels. Now, beer had been brewed and wine had been brewed for centuries before Pasteur, but there was no explanation for a bad vat of beer, okay, or a bad vessel of alcohol. What people didn't know when they were actually producing beer and these kinds of, and wine and so on, was that the yeast that they were introducing in the process of producing alcohol was actually on the grapes or the barley when they added it to the water and started fermenting the sugars, okay? So when it comes to alcohol and we think about wine, well, the sugar comes from the grape. But early on when they were making wine, they didn't add the yeast separately. It just happened to be, unbeknownst to them, on those grapes, okay? So now that we had microscopes that were powerful enough to identify microbes that are actually involved with this fermentation process, this breakdown of sugars to make wine or beer, they were able to see what precise organisms were in the mix, all right? So when yeast is in the mix and oxygen is not allowed to enter those vessels, alcohol is the natural product of fermentation, okay? And this is today how everyone makes beer and wine and these kinds of things. They add yeast. They have a sealed set of vessels that will not allow oxygen to enter, and alcohol is therefore the resounding product. Now what Pasteur is able to show is that there were other bacteria that had made their way into those vessels of beer that were tainted, that didn't taste good, okay? And these particular organisms, in the absence of oxygen, did not ferment the sugar to produce alcohol. These particular organisms preferred acidic products in their metabolism. That's what they would produce. So these are the kinds of organisms that are used to produce now sauerkraut, yogurt, these kinds of things, right? Which people do eat those as well. But this is not palatable to have in beer or wine. Okay. Pasteur is also known for pasteurization, of course. This idea that you could apply heat for a short period of time to kill the microbes that are present in milk, and this would allow the milk to actually be preserved longer. Okay. Pasteur was involved with looking at airborne microbes that can cause infections. He was alive during the time when Sir Lister in England was alive. We'll talk more about him later. Pasteur was active when people were just starting to apply sanitization and aseptic technique to avoid the introduction of microbes in surgical procedures and things like that. All right. Finally, Pasteur was one of the early individuals to pioneer the concept of vaccines, the idea that a weakened form of an agent that could cause disease, whether it be viral or bacterial, could be introduced into a person, not cause the death of that individual, but cause them to become stronger. This idea that you could increase someone's immunity through the use of vaccines was something that Pasteur was looking at at quite an early moment in history. Okay. All right, so we're going to leave it there at this point. A little bit of history up to Pasteur, and then we'll follow up with a little more history next time, kind of get into Cook's postulates and things like that. Okay?